So let's continue on with blood vessels, which are arteries, arterioles, capillaries, venules, and veins. So we've already learned about the heart, and the heart is the source of the pressure that pushes blood through the entire circulatory circuit, away from the heart, out to the body, through the tissues at the capillaries, and then back to the heart through the veins. So leaving the heart are arteries, very big ones, like the aorta. That's the biggest artery you have. And then the carotid arteries and the subclavian arteries that branch off of that, also very large. And then other branches, all the other branches you would have learned in lab that come off of those various vessels are getting smaller and smaller and smaller the farther you go. And eventually, as you get closer to the tissues, those larger arteries are going to branch into arterioles, which are very small. They're not the smallest. The capillaries are the smallest, but they're getting quite small, and they are starting to branch into different areas for bringing blood flow to different tissues. Now, once we get into the tissues, those arterioles are going to branch into a network of capillaries that are in the form of a capillary bed. And that capillary bed is composed of lots and lots of little tiny capillaries that are very, very small. Often only one red blood cell at a time can fit through a capillary, so it's only wide enough for one red blood cell to go through. So really, really small. And this is where exchange of oxygen, carbon dioxide, nutrients, toxins, waste products, ions, everything will be happening between the blood and the tissue. All of that happening at the capillary. Now, as we get towards the end of the capillary bed, those capillaries will start to fuse together to form venules, which are about comparable in size to the arterioles, but they are carrying the blood back from the tissue, heading back towards the heart. So that's why I've turned them blue. Red here is indicating oxygenated blood coming out from the heart, going towards the tissue. Now the oxygen has been used up, it's been depleted by the tissue, so just by convention we make it blue. The blood is not really blue in the venous system, but just by convention we make it blue. So the venules are going to then fuse together to form veins, and these veins would be the size of the things that you would have learned in lab. So all the veins that are coming out of the various body parts, and they continue to fuse, getting larger and larger until we're at the vena cava back to the heart. So the blood coming out of the heart into the arteries, there's a lot of pressure because it's going strongly right from the source of the pressure. So a lot of pressure changes happening in the aorta. It needs to have a lot of elastic fibers. Same thing with like the carotids. If you put your finger in the little dent in your neck up by your chin, you can feel that pulse. That's the ventricles contracting and pushing blood up through those vessels, and then when it relaxes, there's less of a pressure, and you can feel that pressure change happening. So lots of elastic tissue surrounding these arteries that are close to the heart. High pressure, lots of pressure changes. Now, those pressure changes get dampened out the further you go, so you end up with more of a smooth pressure as you get farther away from the heart. The pressure becomes less intense. The pressure change becomes less intense. And also the pressure itself is going to decrease because the source is farther and farther away. So really high pressure here, pressure just continuously going down as you get farther out. Now pressure will continue to decrease as you pass through the capillaries and then back up through the veins. So the pressure gets really, really, really low by the time you've reached the heart again. Now we should note too that the arteries are controlling flow to a tissue. So they control what's called perfusion, which is just delivery of fluid, blood essentially, to a tissue. So if you think of how much blood is going to a tissue, that's how well perfused the tissue is. And you want perfusion to be just right. You don't want too much perfusion and you don't want too little perfusion. If you have too much perfusion, then you can end up with a situation where you have an osmotic imbalance, just too much fluid getting to a tissue, causing swelling, what would be called edema, things like that. So just, you don't want too much fluid going to a tissue. And conversely, you don't want not enough fluid going to a tissue, as would be the case in, say, dehydration, so it would be kind of the opposite extreme. Or ischemia, which just means a lack of blood flow to an area. There you would also have osmotic problems, just in the opposite sort of way, and you would have difficulty flushing out waste products and bringing nutrients and gases to the tissue. So the arteries are able to contract, so constrict, vasoconstrict, or they can relax, vasodilate. When they're constricting, less blood flow to a tissue. When they're dilating, more blood flow to a tissue. And think of like the sympathetic flight or fight response. So if you think of when you're in the fight or flight response and you're wanting to bring more blood to your skeletal muscles, to your brain, to your lungs, things like that, so that you can run away from whatever it is that is threatening you. That blood is going through arteries that are dilated. So you would dilate the arteries going to the brain, to the skeletal muscles, to the lungs. Now, at the same time, you need to have a trade-off there. So you need to reduce blood flow to places like your stomach or your intestines, your kidneys. You're not really worried about filtering blood to make urine when you're being attacked, or digesting your sandwich if you're running away from like a lion or something like that. So you're going to have less blood flow there. So those arteries would be constricted. Now the arterioles are really fine-tuning where the blood is going. So they can constrict entirely and completely cut off flow to a capillary bed, or they can dilate to increase flow to a capillary bed. So each very specific tissue is controlled by what's happening in the arteriole before it. That arteriole can also bypass the capillary bed entirely, and this is called an arteriovenous anastomosis. An anastomosis is just a fusion. You can see that. Let me zoom in on that a little bit more. An anastomosis is just a fusion of blood vessels. And in this case, it's connecting the arterial system to the venous system and just going right around that capillary. So you just sending blood to that capillary. If you think of like like a bypass that goes around the town and skips all the traffic lights, like kind of a shortcut around the town. It's sort of like that same kind of idea. Now for the veins are having a low pressure situation, so they're just sort of passively having blood flowing back up to the heart with venous return. So venous return is how much blood you're returning to the heart through the veins. And they're going to have less smooth muscle because they don't need to dilate and constrict the same way that arteries do because they're not controlling perfusion. But they're going to have more trouble returning the blood because they're also fighting against gravity. So there's going to be some mechanisms to prevent backflow. So let's just compare and contrast our arteries, our veins, and our capillaries. With arteries, like I said, they control perfusion. They have higher pressure because they're closer to the heart. They have more smooth muscle because they're controlling perfusion. They have more elastic fibers to enable them to snap back from the pressure changes. And because of all this smooth muscle and elastic fibers and just general structure of the artery, where it has a more firm structure, so it's a very firm tube. Even when you're dissecting a cadaver and you get to an artery, it still retains its shape pretty well. So that gives it a bit of a resistance to flow, which we're going to get into in our next video. The resistance to flow is just, anything that makes flow slow down, like friction. And you'll just have more of that in an artery. Now veins are for passive venous return. So they're just carrying the blood back from the tissues. They have much lower pressure because they're farther away from the heart. They don't have as much smooth muscle because they don't need to constrict and dilate. They don't have elastic fibers as much because they're not having that same pulsatile flow that arteries do. But they're also kind of floppy. They have what is called capacitance. And that means more of an ability to stretch out, but not to regain their shape. So if you're dissecting a cadaver and you look at veins, they're just sort of collapsed. They don't hold their shape. So because of that, they don't really have a lot of resistance to flow. So that helps to prevent backflow because you just have this low pressure, you're combating gravity, but at least you don't have a lot of resistance, so that's good. And then they also have valves that prevent the backflow. Now capillaries is where we're going to spend most of our time. So that's where exchange of gases happens. We're going to be exchanging fluid, ions, nutrients, all the things that need to be exchanged between the blood and the tissue will be happening at the capillary. And the capillaries don't really have the same wall layers that the other two do. The bigger the vessel, the more developed the wall, which just makes sense. If you have a big vessel, you need thicker, stronger walls. And the smaller the vessel, the less developed the wall. The walls will start to decrease as you go farther out, and as you get to the capillaries, it's pretty much down to just endothelial cells and a basement membrane. So really, really thin walls, which allow for easy diffusion of various substances out of the blood. So water, ions, things like that. Okay, so we've got three different kinds of capillaries. There are continuous capillaries, there are fenestrated capillaries, and there are sinusoid capillaries. So in a continuous capillary, the endothelium is continuous. The endothelial cells are right next to each other. There's no holes or gaps in the endothelium at all. So gases can cross through here easily, water can cross through here easily, so can lipids. Ions can, we know ions can move across cells with various ion channels. So we can get lots of things moving out of the blood and into the tissue. When it's moving out of the blood and into the tissue, that's called filtration. We can also have things moving back from the interstitial fluid, from the tissue into the blood, and that's called reabsorption. So you filter things out that you're trying to deliver to the tissue, and you reabsorb things back into the blood that you're trying to remove from the tissue. So like, let's say oxygen gets filtered into the tissue, carbon dioxide gets reabsorbed out of the tissue into the blood. Now, one thing that cannot pass across a continuous capillary would be these, which are representing proteins. Big proteins and a little protein there. So they're stuck in the blood. They can't get out into the interstitial fluid in a continuous capillary. Now, for a fenestrated capillary, there are little gaps or little holes in the endothelium. The word fenestration means a hole. In a kind of old-time language, if you think of like a castle that has those little slitty windows, those are called fenestrations. And that's sort of what it would look like. Just think of a castle with those little long rectangular windows that you could barely like fit your hand through. That would be what a fenestrated capillary looks like with these little gaps. So all the normal things can pass through here, and they can pass more easily because now there's a little hole for them. Like picture water just flowing through a hose that has little holes. It's going to come out that much easier. So water and ions and lipids and gases can pass through, but now also little proteins can get out. This is going to become important later to follow these little proteins, because now you've got little proteins kind of sitting around the capillary wall on the outside in the tissue rather than inside the blood. And it's going to become important because proteins are attractive to water and solutes, and we're going to be delving into that quite a bit more. Now, large proteins are still stuck in the blood, so they're not able to get through. The holes are not big enough for them. Now, for a sinusoid capillary, really big holes in the endothelium, so these big proteins can get out. It's the same idea as fenestrated, but just more extreme. We have larger holes for larger proteins to get out. Smaller proteins, water, gases, ions, lipids, all of those can get out much easier as well because of that. And again, remember these proteins. Now we've got some big proteins out here along with the little ones, and they'll be attracting water and ions and things like that out into the interstitial fluid. So with a continuous capillary, we can filter just water, ions, gases, lipids, no proteins. Fenestrated capillary, we can filter all of those things plus small proteins, and then with a sinusoid capillary we can filter everything, including the large proteins. Okay, so for Bastles Law, This is regarding blood flow. What governs the flow of blood through your blood vessels? So what are the forces that cause blood to flow either quickly, strongly, or less strongly, so a slower sort of flow? And the equation is flow equals change in pressure. Delta P means change in pressure. Delta always means change, and P, pressure, divided by resistance. So flow and delta P are directly related to each other. As delta P increases, so does flow. If delta P decreases, flow also decreases. So they move together. Flow and resistance are inversely related. So the higher the resistance, the lower the flow. And that just makes sense. If you have low resistance, then the higher the flow. And then you put them all together and you get the total flow of blood through a blood vessel. Now we can take this away, and if we look more deeply at resistance, a lot of resistance has to do with just friction of blood on the side of the blood vessel wall. So just picture, here's the inside of the blood vessel, so here's the lumen, and right in the center of the lumen, the blood is completely unimpeded, it can just go. But along the sides, it sort of gets dragged a little bit by friction on the sides. So the bigger, we have this great big vessel, we got a lot of blood that can go right down the middle without getting stuck along the sides, and more of it, or less of it, will be stuck along the sides. So the bigger the vessel, the less resistance we're going to have, just in terms of how wide it is. Also, the shorter the vessel, if you have a really big, wide, but short vessel, not a lot of chance to get friction. If you have a really narrow, long vessel, lots and lots of chances to get friction, so it would have a higher resistance. Just picture yourself walking down a hallway, like a big crowd of people walking down a hallway, and the hallway would represent the blood vessel and the people would represent the blood. So if you have this like long skinny hallway and a certain amount of people are trying to get down that hallway, a lot of you are going to be kind of pushed up against the walls and kind of dragging and not being able to move so fast, and the longer it goes, the harder it's going to be to get all those people down that hallway. But if you have just this real wide, short hallway, everybody's just going to be able to pass right through. So it's the same idea with blood. So if we take resistance, we can think of resistance as it relates to the radius. And if you remember in like geometry class, the radius is half the diameter of a circle, which is essentially the blood vessel here. So resistance is proportional to 1 over radius to the 4th. So it's inversely proportional to radius. So the bigger radius to the 4th is, so the wider the vessel, the lower the resistance, which is what we just said. The wider the vessel, the less resistance we have. The smaller the vessel, the less radius we have, the more resistance we have. And it's an exponential difference, so it really makes a big, big difference. Now, if you think of flow as being opposite to resistance, and resistance is opposite to radius. So resistance is opposite to both radius and to flow. That means that flow is directly related to radius. So just a little mathematical gymnastics here. But it sounds confusing when you put it that way, but if you just think about it, the bigger the radius, the stronger the flow, which just kind of makes sense. Now, as for delta P, we've already talked about the pressure being highest at the heart and the aorta, which would be the beginning of the circuit, and then getting much lower as we get out through the veins, the farther away from the heart. So at the aorta, a typical pressure, just a general pressure, and everyone would be slightly different, is 100 millimeters of mercury of pressure at the beginning of the aorta. That's going to decrease pretty rapidly, and by the time we get to the end of the arterioles, it's down to about 35 millimeters of mercury. That's a delta P of 65 millimeters of mercury, which gives you a very strong flow through the whole arterial circuit. Now from the arterioles, we go into the capillaries. So the start of the capillaries would be 35 millimeters of mercury, because that's, picks up right at the end of the arteriole. And by the end of the capillary, it would get to about 18 millimeters of mercury. Now, this is just an example. If we have vasodilated the arterioles to get more blood into the capillary, we're going to have a really different set of numbers here. or if we have severely constricted the arterioles going into that capillary, we're going to have, again, a different set of numbers here. But let's just use this as an example. So here in this example, the delta P is 17 millimeters of mercury. And that's an okay delta P. It's not that much, but it's also a very small distance in a capillary bed. So we'll have a fine amount of flow. It's not a strong flow, but it's enough to get the job done. Now, once the capillaries at the end of the capillary bed, they start to fuse into the venules and into the veins, and by the time we get back to the vena cava, we're down to about 2 millimeters of mercury. It's a delta P of 16, so not much of a delta P. It's also over a very long distance in some cases. So if you think of going from like your toes, a tissue in your toe, all the way back up to your heart, that's a long distance with this little delta P. So the flow is going to be pretty weak, and that's why veins need to have valves, and it's good that they don't have a lot of resistance. Alright, so that does it for this video. Our next video we're going to be talking about filtration and reabsorption. Filtration, we already said, is where substances go from the blood into the interstitial fluid, so we're delivering to a tissue. The things that are going to affect that are hydrostatic pressures, which essentially is blood pressure compared to the pressure of the interstitial fluid, and then something called colloid osmotic pressures, which has to do with what we said before about how proteins help to attract water, ions, solutes, things like that. So whether we have these proteins, which are colloids, colloid just means a fluid that has protein suspended in it. So are these proteins in the blood? Well, yes, there's a lot of proteins in the blood. And are there proteins in the interstitial fluid? And the answer is sometimes. So in this case, yes, a little bit of proteins out there in the interstitial fluid. In this case, yeah, quite a bit of proteins in the interstitial fluid. In this case, not so much. So we're going to look at those various pressures in the next video. And we'll also look at reabsorption, which is just the opposite of filtration, where we're having things go from the interstitial fluid back into the blood, also relying on those same pressures, but in differing ratios. So that'll be the next video.